Honorable Speaker.
Thank you, Dr. Palak ma'am. Respected Professor Anamik Shah sir, respected faculty members, and dear participants. On behalf of LM College of Pharmacy, I heartily welcome today's expert speaker, Professor Anamik Shah, and all participants to this 11th webinar on a lecture series, Science of Science in Spotlight. So where we invite speakers across the country and from the foreign countries also to enlighten the participants about the research methodology, the research ethics, and the updates in various areas of pharmaceuticals and pharmaceutical sciences. So for the today's lecture, we have requested Professor Anamik Shah sir, who is a renowned scientist, renowned academicians and administrator, specifically in his area of interest is organic chemistry, and specifically in pharmaceutical chemistry. He has done a good work and he has a good number of publications in very renowned and peer-reviewed journals also. So we are lucky to have him as an expert for this lecture series. I'm sure the topic which he has chosen with its case studies or the examples of its own will surely motivate us or inspire us. So being in the academic institute, you can also do the good research, or I would say the research which would be notified by or noticed by the world over. So I again heartily welcome Professor Anamik Shah sir and thank him for accepting our invitation. And I'm sure all the participants will enlighten by the sharing of his wisdom, words of wisdom, or the knowledge with you all. Thank you. Thank you, Palak ma'am. Are you there?
Palak madam, are you there?
I think she has connectivity issue. You can take it forward. Tejas bhai.
Yeah, okay. So thank you, sir, for giving this welcome speech note for this 11th webinar. Now I would like to invite our chairperson of the session, Dr. Dipen K. Sureja. Sir has been working as associate professor at LM College of Pharmacy in the Department of Pharmaceutical Chemistry and Quality Assurance. Sir has been working in the field of medicinal chemistry, organic chemistry, and computational chemistry. And his research expertise includes design and synthesis of biological evaluation of novel heterocyclic scaffolds for anti-cancer, anti-malarial, as well as sir has been also working on tuberculosis as well. So with this very brief introduction, now I would like to invite Dr. Dipen K. Sureja to chair the session ahead for the upcoming events as well.
Thank you, Dr. Tejas. Am I audible?
Yes, sir, you are audible.
Am I audible?
Yeah.
Yes, sir, you are audible.
Yes.
Yeah, okay. Thank you. Good afternoon, everyone. So today I feel honored and privileged to introduce an eminent personality, Professor Dr. Anamik Shah. He did his schooling at Saurashtra High School and S.V. Virani Vividh Lakshi Vidyalaya Rajkot. He completed his BSc in the year 1975, MSc specialization in organic chemistry in 1977, and PhD in medicinal chemistry under the guidance of late Professor V.M. Thakur from Saurashtra University, Rajkot in the year 1983. He worked in different capacities like as a lecturer, reader, and professor at Department of Saurashtra University and has a total of 33 years of academic and research experience. His area of research includes drug discovery of anti-HIV drugs, anti-cancer agents, anti-TB drugs, aromatase inhibitors, and non-nucleotide reverse transcriptase inhibitors. He has also contributed significantly to the field of synthetic organic chemistry, analytical method development of active pharmaceutical intermediates, analytical environmental chemistry, natural product chemistry, and isolation and screening of marine-based natural products. He guided 70 PhD scholars and supervised numerous MSc students for their dissertations. He is also a recipient of more than 35 prestigious overseas fellowships for pre-doctoral, doctoral, and postdoctoral work. He has more than 205 international and national publications in peer-reviewed high-impact factor journals. Additionally, he has contributed to several book chapters and has 7 patents to his credit. He has done incredible research work as the total citation of his research work is more than 9,000 and has an H index and I10 index of 50 and 178 respectively. He is a recipient of a number of projects funded by various international and national agencies like NIH, USA, DST, DBT, UGC, CSIA, DAE, and MSME worth 25 crores Indian rupees. He has also completed several pharma-related projects with Alembic, AstraZeneca USA, Claris Life Sciences, Dava Research Foundations, and Piramal Life Sciences. He is also a founder of the Center of Excellence in Drug Discovery, National Facility for Drug Discovery, NFDD complex at Saurashtra University. He has been invited more than 50 times at national and international conferences to deliver lectures and chair the sessions. He has received many awards for his excellent academic and research contribution. He is also a recipient of a Lifetime Achievement Award of 37th Indian Council of Chemists, ICC. Currently, he is the editor and reviewer of many reputed journals like Journal of Cell and Tissue Research, Medicinal Chemistry, and Indian Journal, Journal of Basic and Applied Pharmaceutical Sciences, Brazil, and Heterocyclic Communication, UK, to name a few. At present, he is serving as a trustee, chairman, board of director, and president at renowned trust and organizations. He has also served as a vice chancellor of the prestigious Gujarat Vidyapeeth for almost 6.5 years. During his tenure as a vice chancellor, Gujarat Vidyapeeth was accredited A grade by NET. With this brief introduction, I invite Professor Dr. Anamik Shah to deliver a talk on research and innovations in drug discovery at academic institute and hybridized collaborative models with pharma industries. Few case studies and lessons to learn. Over to you, sir. Anamik sir. Thank you.
Thank you very much for the long introduction. But Dipen is personally also known to me as he belongs to this region and one of our family friend. I am very happy for the invitation given to me on this occasion and this special lecture series at very prestigious LMCP, which we are sometimes calling it Makkah of pharmaceutical industries of Gujarat and also other parts of India. I am highly obliged for the invitation from one of our very close friend and principal, Professor Mahesh Chhabariaji, who is a wonderful researcher and also academician and administration and he was on the board of Gujarat Vidyapit for almost 3 years with me, and we have worked together. I visited LMCP several times from the days of Sisu sir, Goel sir, R.K. Goel sir, and many others. And currently also, I will, I am taking opportunities to visit, and I'm very happy that the LMCP has now a state-of-art facility contributing in a very finest matter at the uh, Ahmedabad, and many small and pharma and medium enterprises are benefited by the research work and output from the researchers and students of LMCP and faculty members. I just want to start my journey, and this journey is a beginning of from a small facility at Saurashtra University, which were ultimately culminated in a very big, large facility with the help of philanthropists, with the help of government of Gujarat, and with the help of Department of Science and Technology, and almost many of the funding agencies. Each has contributed very well, and also my national and international collaborations. But let me tell you that this is a trailer of evolution of our team members, and also the participants who are in this webinar, they need to evaluate at the end of the presentation. In fact, my presentation is that in ever-going pharmaceutical science, there are enormous opportunity. If catch at the right time, the sky is the limit. But research, innovation, and entrepreneurship is boundaryless phenomena. So even if you are trained in masters on a particular specialization, you need to go out of boundary time or boundary line so that with multidisciplinary, interdisciplinary approach to emerging area will lead you to the doorstep of success, but to be failure and record date on this is also data on this is also a learning tool. So I think that I will begin with some of the important topic one by one, and at the end will lead to something which will be a take-home message for you. Next slide, please. Somebody can, yeah. Friend, this is a wonderful photo of Mahatma Gandhi, Sardar Patel, and lady, that is Dr. Sushila Nair, who was Chancellor of Gujarat Vidyapit, but she worked with uh, Mahatma Gandhi, and the young person whom you see is Dr. Y.K. Hamid, who was the founder of Cipla Limited. And Gandhiji has wrote a one-line message to the Cipla company, which was started at that time. And you'll be happy to see that what he has written, that I was delighted to visit this Indian entrepreneur, M.K. Gandhi. So you can just very well understand that entrepreneur sabda, word, and self-reliance was very much in the heart of Gandhi, and Gandhi was with the modern changes, but in decentralized manner, which Cipla has done wonderful with its commitment to the poor people for the affordable medicines in the third world countries and many others. So let us start our journey from this photograph to the next one. When we're talking about the collaborations, what are the type of collaborations possible? I think that if you enlist from new chemical entities, intermediates, modified key starting material, contract manufacturing, research, route of synthesis, economical viable route, process chemistry, green route and green chemistry, clinical research organizations, catalysis or without catalysis, recycling of catalysis, chemistry and chemical engineering labs, coordination and orchestration, pilot plant operation, scaling up operation, synthetic route, and in modern trend, it is the quality by design, starting from the equipment itself. And large and tall industry plants to the very small and tiny plant of flow chemistry, then diagnostic agents and medical equipments. So these are where pharmaceutical science, pharmaceutical chemistry, process chemistry, catalysis, environment, sustainability, all are covered in this possible collaboration. And I will just go for the next slide, that how from a small few, what we call it, round bottom flask and condensers, and that too not the quick fix type. We have grown little bit by little in last one to a very good institution heritage at Saurashtra University. This is the academic institutional framework in India, and we are aware about that. So it is just, we know that the first thing I just want to mention, that the Institute of Chemical Technology was the unique model where the uh, the director of the institute has given mandate to all teachers that your salary should come from your consultation, and you have to multiply your salary by getting more and more consultation. And we know that it's the unique institute. Now it is Institute of Chemical Technology, Matunga, where Dr. Maselkar, Dr. M.M. Sharma has developed the entire legacy, and also Dr. G.D. Yadav. Next, we know that before independence, several CSI labs were started. And the goal of CSI labs was to augment all facility under one roof. If you see the CDRI, CDRI is covering everything which is related to drug discovery. And even if many drugs has not come out with the with the CSI labs, uh, the main thrust was to train the human manpower. And we know that in major pharmaceutical companies of India and abroad, the CDRI people, the IICT, NCL people, they are across the world, and therefore the importance of that. We have several institutes like Department of Atomic Energy, Department of Earth Science, Ayush, ICMR, and many other private pharma research centers at that time. Multinational companies has put their effort to have wonderful centers of research. Our journey was beginning from 1990 when we approached CDRI, especially for the biological activity of cardiovascular compounds. We have developed several of ingredients, novel structures, and we want to work on that. And first time, without taking any money from CDRI, has tested our hundreds of compounds. But later on, we have very meager facility at that time in the department, and department has no UGC fund, and UGC was not supporting very well at that time. So we received some communication from multinational companies like DuPont, Merck, R.J. Johnson, and Specs, Biospecs, and later on AstraZeneca, that can you prepare small molecules for us? We have no parallel synthesizers, but our students were parallel synthesizers. We have only one research lab, and students were working in different uh, streams, morning batch, evening batch, night batch, and they are working, they are putting their reactions and developing small molecules, and we are supplying to these people. And then we earn lots of dollars out of that. And from that, we built up two more research lab, one library, and one seminar hall from AstraZeneca's money. And then with this work, we learned that how to prepare a compound with adequate purity, how column chromatography will help to achieve the purity of compound, and many things we have learned without any analytical or instrumental support at that time, because we were supplying the pure compounds and structures were determined by some of these companies. Later on, Ranbaxy come forward in late 90s, and they wanted academic tie-up to develop certain specific molecules for anti-infective. But this experience of getting dollars and this distributed to the guide, to the students, gave a new vibrancy to the students to do lots of things, and they have clicked many ideas how to get small molecules with less efforts on the synthetic methodology, and we can have a diverse type of molecules which compound library may be of interest to these people. So this was our initial experience in the early years. But next time, when we have a proof of concept and other interests shown by the foreign pharmaceutical companies, later on Indian major came to for collaboration. So Alembic wanted anti-infective and antibacterial compounds, and they come with a Department of DPRS project, Department of Pharmaceutical Science under DST, and we got a 60 lakhs of project. Dabur Research Foundation was specific on cancer work, and they wanted the cancer activity to be prepared by us. And we prepared several coumarin molecules, benzopyran molecules, as well as we prepared some indolinone derivatives, because at that time, a Sugen Life Science, a very smart company, has come out with the first molecule, very simple molecule, getifinib. And now we know that in cancer, there are more than 150 compounds of NIB series, and many of them, half of them are in the market now in last 25 years. Similarly, Ranbaxy Laboratory also gave some funds, and then came Pyramid Life Sciences. I will give you some idea that how these companies were interested, but simultaneously for good paper publication, and when facilities were not available with us, we felt and our dictum was one word dictum, that when you don't have any facility at your place, the facility, either instrument facility or biological activity of the rest of the world, belong to us. So you connect and collaborate, you communicate and get the thing done from them. So this is how what we were thinking about. I have written the logo of Claris Life Sciences, and the story will come later on, how we work and what we contribute to the Claris Life Sciences. So this is the first half of 2001, but simultaneously we did collaborations. So we collaborate with industry and academia, small pharma companies, and open source drug discovery platform like OSDD of Department of Science and Technology for tuberculosis, and the target was absolutely novel. It was GLMU target. And then we also worked with the Lilly, and we worked with the TAF for anti-tubercular front. We'll come to the BRNS work, but we also started in 2016 when I left this, but work was started for molecules, and it is for drugs for neglected diseases. And it was a platform where the scientists world over, multinational companies and academicians work together for the drugs which are abandoned by the multinational companies and profit-making companies, but the poor people and affordable people need it. So the right-minded people worked on that and developed certain molecules, and they are in market. So during this span, there were many international collaboration and national collaboration era. It was beginning. In national collaboration, we have a bioinformatics collaboration with Professor V.M. Kulkarni, because we didn't have the facility for Schrodinger at that time. The Defense Institute of Physiological and Allied Science, they are working on anticoagulant compound on monkey, and we were preparing design compound for them. Then in Mysore University, there was signal crystal XRD facility. In Bombay College of Pharmacy, many of you are knowing Professor Coutinho. We published so many papers of biological activity and bioinformatics. And for natural product, we have a collaboration with JSS College Ooty. But simultaneously, uh, we have also tried to have collaboration from the Belgium and Greece and Italy and Hungary and Japan and Taiwan. And since we are engaged with the Indian Society of Chemists and Biologists, many of our friends who were coming from overseas, they met our students in ISCB conferences. They have seen their posters, their enthusiasm, and their work culture, and their contribution in the preparation of the conference, and their keen interest in variety of medicinal chemistry topics and subjects in which they wanted to work a lot. So this has given us a good direction, and we, from 1996 to 2014, we are able to have a wonderful networking of different collaboration on anti-tubercular, TAF USA, anti-HIV with Catholic University, Professor Clark. And Professor Clark is one of the pioneer in the click chemistry. And he has more than 300 papers on click chemistry. And we know that this time Nobel uh, prize is given to the scientist who has worked on click chemistry. Then we have a Professor Motoasi from Meiji University, Professor Molnar from Hungary, Albert St. Georgi Medical University, and Professor G.M. Petros Garagli, who are working on our MDR reversal agents, where we have given a proof of concept of some of the wonderful molecules. We also tied up with one time this our prestigious Indian award, Shanti Swarup Bhatnagar awardee Professor Virendra Pandey, who was from Department of Atomic Energy, migrated permanently to ASA. And similarly, we had a Professor Roberto Lordo, who worked in Italy. All this collaboration, we have never met. Sometimes we have never seen faces, but in some conferences, 1 or 2 person we have met. But otherwise, everything was by correspondence, by postal work, as well as when internet facility came, everything was done by internet. The conferences has given us a trust, and our many talented researchers were gone for pre-doctoral, doctoral, and postdoctoral work without spending a single rupee from their pocket or their parents' pocket, and they were uh, migrated for 1 year to 3 or 4 years for this program. Belgium, Greece, Italy, Hungary, Japan, Taiwan, USA, UK, Portuguese, Ireland, and many other countries on under the Erasmus Mundus European Union Fellowship, Eurex, Marie Curie, and Humboldt Fellowship also. So this is how many things has happened at that time. These are few people who have been back to homeland. Some of them have retained in USA, but they had got this Alexander Humboldt Fellowship, Marie Curie Fellowship. So more than 25 students have got this benefit. Similarly, in Leuven, Oxford, and other places, University of Göttingen, they got the position. And many of them visited in Taiwan for anti-cancer work with Professor San Lon Sus. Yes. Academia Sinica is one of the premier institutes working on 4 different type of disease processes. Similarly, some of our students has got this various fellowships. Humboldt also, Atul Manwar now back to India, and Pratik Ambasna was for 10 months in Belgium, but he prepared 5 papers and 1 review article. Hetal Patel, after her master degree, she got the best PhD thesis award at Rutgers University, and now looking after the regulatory affairs of Pfizer vaccine for COVID-19 for the entire Europe. And her husband has first time got NIH grant, converted a synthetic organic chemist into the viral scientist and virology for non-nucleoside under the NIH grant. So during this process, our students have got capacity building in catalysis, microwave assisted organic synthesis, solid phase peptide synthesis, cancer pharmacology, biocatalysis, flow chemistry, bioinformatics, medicinal chemistry, and many others. So this is how the expertise has gone toward indigenization. Those who have come back, they are helping their companies. So far as my data is concerned, our more than 25 students has their own API company. 15 may have intermediate manufacturing. Few are recycling costly catalysis like ruthenium and other things. Many have started their plastic and polymer industry and tiles and other things. And they have gone to formulation industry also. So this is how. And in Rajkot, due to this work culture, the DN Vice Chancellor decided to establish a new Department of Pharmaceutical Science, which was initially supported by Department of Chemistry by all means, by faculties, by instruments, by all facilities they are shared. So this is a center which was built up by the philanthropists, money, Saurashtra University money, and 35,000 square feet, 3-floor building, and many laboratories and instrument facilities and this. So this was the exemplary thing, because at that time, very few funds were available, but we were able to get 10 crore rupees in the first phase. In the second phase, we have 13 crores. And later on, you can see that different type of instruments were and facilities were in this center. We have 8 fume hood laboratories, then several parallel synthesizers, rota vapors, and preparing for the small molecules. Then we have a single crystal X-ray diffraction facility where we have identified 200 crystals structure of small molecules, and more than 10,000 NMR supported by our technicians for getting NMR to the researchers of entire India. So more than 50 universities and 250 researchers were got at a very low price NMR data done. Similarly, we have thermal stability chambers, DSA, elemental analyzer, microwave synthesizer, UPLC, then HPTLC, flash chromatography. And this is the service available at that time. Right now, due to paucity of the technician person, the facilities are idle, but I hope that the current administration and government may take notice of this enormous facility created by hard labor by the research scholars and faculties and Department of Chemistry and the PI uh, of the entire project. So these were the various services. You can later on look to the things that how instrument can be very vital to support the small pharma companies and help in a great way. So you can just see that this is the entire instrument support. We gave support to 50 institute and 200 research scholars. We started a PG diploma for sophisticated instrument center for 1 year where 8 different instruments were taught to the students and they have to do a dissertation on that. So they were ready to utilize by the industry immediately if they want any analyst or instrument operator. And more true, beneficial was that all our master students of the fourth year, second year, they were given 100% dissertations, either in instrumentation or in the synthetic drug and preparing at least one drug and analyze it thoroughly and investigate different routes of synthesis also. So this is how the quality of research and training of the research student has got a very good value between 2005 to 2016 and onwards. And pharma companies were ready to take if the students were from dissertation students were from Saurashtra University or especially from the Department of Chemistry and their PhD work. What model we had adopted, because government will give grant, but they will not give you the people. So we adopted this model, and I think that any research model or instrument support center or center of excellence can adopt this model, and this can be a very much workable model. We worked successfully almost 7 to 8 years, where we recruited top retired professionals from pharma industries. We recruit full-time PhD experience from industry with experience. We gave the internship, intern full charge of instrument. They were assigned work of in-house and outside samples. We are giving them fellowship support or income distribution. We are tying up with the small pharma companies and we are applying for the grants. We wrote and prepared new patents, non-infringing patentable process with industry, filed and granted. And we also helped in troubleshooting in process, catalysis, purity, and route of synthesis also. So what lessons we have learned in this is the selection of ROS, purity of substances, impurity identification and characterization, green chemistry approach, scale-up operation, recycling and recovery of catalyst, time schedule in tune with industry and deliverable to the identified, and analysis of the results to the needy. So uh, this is what our experience in nutshell I have shared with you. Now I will share some of the work which we have done with the pharma companies. And from sitting at Rajkot, we worked and we built up with teamwork with the Piramal Research Center. And we were working with their MedChem team, IPR team, biology and pharmacology group, cancer pharmacology group, synthetic group, bioinformatics and chemoinformatics group when we had worked with them for a very specific molecule which were for the cancer. So in our university, we have developed discovery of DP7 as a multi-drug reverter in cancer. We worked with alembic 17 structures as antibacterials. KUQ18 was identified as promising lead at Piramal Research Center. Two molecules were for TB. It has reached up to GKO model by TAF, YN9 and NM140. Coumarin compound were very active with Dabur Research Foundation. We give a cheapest technology and most competitive technology in competition with the China of 5 keto acid to the Claris Life Sciences. And the sad story is that we have not received the royalty grant for which we, Saurashtra University, was entitled, because this keto acid, when formulated, the value of this keto acid becomes enormous, but somehow there was a weak tripartite document, and therefore we could not avail this. So there is a warning bell while framing the legal documents with the small to big pharma companies. Then we also worked on non-nucleoside reverse transcriptase inhibitors. This is the KUQ compound, and these are the cleaning results. This was with, as I said, it was with the Piramal Research Center. These are the profile as TNF alpha inhibitor and summary of KUQ. So it was up to zebra fish toxicity. Next. You can go to the next page. Hello. Tejas, you can go to next page. Yeah. So you can just see that it is up to zebra toxicity. And this is the paper which was published recently in SES Omega. And this is our compounds where very good activity was given. Similarly, there was a wonderful molecule DP7. Basically, it was a dihydropyridine, but the design was such that its calcium channel activity was reduced to 0. And we worked on that. Similar molecules, a book chapter is already there in 2008, but the molecule was upgraded time to time with continuity. And you can just see that a single molecule and related compound has come out with 15 beautiful top cited journals. These are all chronology of publication of a single molecule. So when your molecule is there, don't leave it, do it for several purposes, and see that the entire biological spectrum is studied very carefully, and therefore it can achieve very good publications out of your small molecules. We got a patent also in multi-drug resistance reverting agent. It was with NCL Pune in collaboration. The bioinformatic work was done by Professor Kitunia. The molecules were synthesized by us and further tested at Dhiman Sarkar's lab at Pune. So this is how, this is the DP7 structure, this is the crystal structure, and this is the patent we have taken on this molecule. Now, I just want to say a little bit about the NIH grant. Why I want to say that, when the molecules were given just to screen it, they were giving wonderful activity. So we wrote a NIH grant, and to write a grant of NIH, it's a big issue, and very few people can write NIH grant application. But we decided that we want to get NIH grant. We submitted, and we were rejected. That your application is weak, and there is no rationale for non-nucleoside reverse transcriptase inhibitor for which you are going. So we introduced a bioinformatics person, student of Professor V.M. Kulkarni, staying in USA, and further we resubmitted the grant. And as you know, that NIH grant is always evaluated by not single person or two person, by 27 top medicinal chemists, scientists from pharma industry, from NIH, from multinational pharma companies. And fortunately, we received the grant and we worked on this molecule. But one of the interesting take-home message is that when we didn't have a bioinformatic person, the molecules which we predicted by our intuition, they were much more active than what we synthesize after the introduction of the bioinformatics. So sometimes the reversing things is also happen, and because it is a science and truthful science, so you cannot claim that every time whatever prediction is there, it will come as a truth. So this is the learning lesson for the project writing, failure as well as success. Now I just want to say about the process chemistry of keto acid. Uh, the keto acid were imported by China, and Claris has a product which is known as ketolog. Now this ketolog is a mixture of 5 water-soluble amino acid, which is taken in the form of tablet, and it reduces the occurrence of what we call it the blood transformation for the kidney patients. So this was a oral tablet, and for which the China supply was given to Claris, and it was little bit costly at that time. Claris, under drugs and pharmaceutical research project, supported by 70 lakhs from government and only 10 lakhs from Claris, within 2 years we come out and developed by the same adduct 4 keto acid, where there was a uniqueness of process, same starting material. Last 2 steps are in water, salt formation also in situ with adequate purity. Purity was 99.95, acceptable to the industry and pharma standard, and few steps of unit process and operations were involved. So this is how we develop this keto acids, and now many people are making this keto acid at in Gujarat and other places, and they are formulating it for this. But the transfer of technology, it has replaced Chinese import. We learned how to work with the industry with attitude, aptitude, and adaptation. We require time-bound delivery and confidence from the industry, mutual trust and collaboration, and confidentiality. So this is how we learned a lot from this. I just want to cite another interesting project which we have taken up, and every college and academic institute should take up, and that is to preserve their old molecules of master degree, MPharm, and PhD in medicinal chemistry and pharmaceutical chemistry. It is very easy to handle these molecules, store it in a good and cool places, and whenever you test it further, check the TLC before using for the biological activity. So we created our own library of our own compound, simultaneously PhD guides from all over Gujarat and other places, and it was known and funded by Government of India to create the facility for preservation of molecular diversity. So these are the more than 12,000 compounds already available at Saurashtra University, and anybody will request Center of Excellence at Rajkot, they can get it for variety of biological activities with the structures. I think that they have, after leaving that, they have saved the database and the code name of the molecules, and it can be from the theses and from the people who have supported that. I think that this is time is 2:28. So somebody should tell me that how much time I have to I have left. Tejas,
Sir, yeah, sir, you can continue.
nahi, but you give me the upper limit.
Yes, sir, we may have 15-20 minutes more.
15 minutes. Okay, that's fine. Okay. Yeah. Now, see, I am giving this lecture in different compartments. So sometimes you may feel that there is a loss of the connectivity, but what I want to show you is how we can take the different approaches for the research. One of our students, whom I have captured from Dr. Rakesh Rawal, your neighbor at Department of Biotechnology at Gujarat University, and he was interested in doing some bioinformatics work, and we had just procured the Schrodinger suit for bioinformatic tool. So Farah Sheikh came to me. He was given a Fulbright fellowship under the project. He remained with us, prepared 15 papers, and then he joined Macau University for his PhD program with Chinese professor Shirley. And meanwhile, he was invited by Oxford professor for 6 months. But we know that computational target prediction is a very interesting learning, and there are the many tools of machine learning which are coming for the target prediction methods. This is the graph where we can see the different methods, but I just want to pinpoint what Faraz has done during his PhD work and at the University of Macau. So these are the various methods of Zhang, structure-based, ligand-based approach, ligand and structure-based approach. Then PASS is ligand-based, and we have C also. So these are the various known targets on which people can do lots of bio and chemoinformatics works. And from 2006 to 2020, these are the methods people are using. Some are open source, some are paid, and therefore a new open source drug discovery platform was necessary as per Faraz and his guide. So they come out and they studied the limitations of the existing method. I'm not going into the details of that, but you can just see that the limitations of C in Journal of Informatics, it is given. You can read at leisure. You can circulate my PowerPoint presentation to all participants, and there is no issue on that. So what Faraz and his team came out and his guide, they started leak the map, and they identified the 17 different human targets: kinase, transferase, beta secretase, hydrolase, anticoagulant, carbonic anhydrous, up to HIV, tuberculosis, HCV, influenza, diabetes, and Helicobacter pylori. So this is how they worked on that. So this is the binding fingerprint region where some work is being done, and you can see that there is a hydrophobic portrait, ionic and protein anion, and how the molecule fits in. But Faraz and this LeakD map workflow is like that. You prepare the query of compound in Smiles. It is open source, and anybody who is in M-Pharm, who is in B-Pharm, can take up the structure of molecules, put it in the all 17 target classes, and then you will get the class specific predictions and activity predictions, and you will also see the binding model prediction also. So lots of people in their world is using this, and it is a known as random forest algorithm. So training of class-specific ligand binding activity prediction models were searched, and these are some of the case studies of HIV drug target, but I'm not going into the detail. You can go into the 2019 SCS MedChem paper, but I just want to show that this identification, many countries have worked on that, and you can just see that the United States, more than 4,000 people have worked on that. In China, 2,000. In India, 2,000 people. In Spain, 1,000. In Germany, more than 1,000. In Brazil and other places, Taiwan, more than 500 people, including Canada. So this is the contribution of open source drug discovery and the basic training by Faraz Sheikh, Nirali Desai, and Sully. You can take this bioinformatics paper of 2021, and I can share the link. So those who are working for the project work at B.Pharm or M.Pharm level, they can take up this job and interesting results will come with the diversity of the structures. I just want to show you the structure of our patent, which is on the what we call it as the multi-drug resistance tuberculosis. So this patent was taken as with the Dhiman Sarkar and Dr. Coutinho. So how Indian scientists can collaborate in the different domain and achieve the good publications as well as patents. That is what I just wanted to show with this patent. So this is the publication of that paper, and you can just see that novel pyrimidine scaffold was published for the dormant tuberculosis basically, and we know that we have no drug for the dormant tuberculosis, and therefore these molecules and class of compound they were taken patent in different countries under PCT. I just want to show something which is very important in current this sustainability and environment aspect. And one of our students who got trained in especially in the Ireland, and he was one of the consultant of Johnson and Johnson. So he worked on the Mitsunobu coupling under superheating and flow chemistry. And flow chemistry, as you know, that there is an input of different starting materials and solvents and catalysts. On other side, there is an outflow of product uh, um, um, and unutilized materials, solvents, and other salts and other things. But instead of 3 or 4 or 5-story or 7-story multi reactor giant plant in the small office room where you have all this flow chemistry, flow reactors, tiny reactors. They are working very well in the less hazardous conditions, and daily production can come out from the small room around 10 tons per day, and you don't require the huge giant manufacturing plants. So the next phase of time will come for such type of flow chemistry work, and which will lead to this. So these are the two papers we have published earlier. Now, very prime universities, IITs, and other people, they are comparatively 20 years late, 20 years late than China in the flow chemistry, because migrated Chinese scientists are after flow chemistry, and they will come with the much cheaper methods of manufacturing from drugs to many other products by this way. These are the few important publications which were done by the collaboration at Taiwan, and it was absolutely on anti-cancer and cross-linking DNA and topomerase 1 and 2, and these compounds were modified nitrogen mustards as well as phenanthrene and pyrrolophenanthrene and anti-tumor indoles. We developed few papers on this potential P glycoprotein inhibitors, and also we worked on the 1,4-dihydropyridine based triazole derivatives, where we had a in silico and in vitro study also. And this is how many things have come out. and anyone can see these publications or ask us to send a copy of that. Some TB work we have done earlier, where coumarin compounds 7-member system was clubbed, and they were giving the Mycobacterium tuberculosis activity. So these are the work which were done on the few things. These are the MDR cancer reverters, and I can say that the group of mine working at Sauras University, they were the first pioneer in entire world to show that the dihydropyridines can be a good MDR reverter, and many lead molecules can come out from that. So these are the few publications on that. These are some other publications. I just wanted to show you a little bit, but okay. Some bioinformatics work we have done in collaboration with many people, and these are such papers. And I just want to show you the deuteration. In last couple of years, many of drugs were deuterated, and it is believed that the pharmacokinetics and pharmacological profile of the deuterated drug may have better pharmacological profile. To study that and to understand the mechanism of how this deuteration can be carried out, we took some molecules like valproic acid and mesalamine and even lamotrigine, and we have deuterated that, and then we nuclear and radio isotopes, and we came out with some good publications from that, and we successfully deuterated by using palladium and platinum mixed catalysis. These are the patterns already, somebody has already alerted that these are the patterns, but some patterns we have done, which is on the tricagellar and other products at the national facility at that time, and they are filed. So I have an opportunity to meet good people and get good advices and support. One of them we have lost very recently, Professor C.L. Ketarpal. Professor M.M. Sharma has also visited our facility at that time. And this is Professor Sukhdev, a doyen and godfather of synthetic methodology, who is now retired and at the age of 93, still writing the effect of uh, the kitchen uh, the kitchen products and what we call it spices and its medicinal use and how the medicinal chemistry is involved in that. So these are the people who are permanent inspiring for us. And I think that I've taken much time, but I again thank you to all of you for giving me this opportunity to share a few things with you. And as I have told you that the take-off message is that there is no boundary for our success, and there will be always accidents and failure, but without accident and failure, there will not be success. And therefore, success will begin and it will be always followed to the failure and accidents. So wish all participants a very good future. And I thank again Dr. Mahesh ji and our all friends, and especially Dr. Ketan Rank for inviting and coordinating me, and the person who has helped in putting up these slides on behalf of me and relieving me from that labor. So thank you very much, and good luck.
Thank you very much, sir, for your good lecture. You have covered many topics in the lecture, and you have shared your experiences and contribution with a few case studies. So, sir, do you have time? We have few questions with us.
Sure. No issue.
Yes. So, first question is from Drihan Patel. What is the difference in invention and innovation in a broad way?
See, there are multiple definitions of both. And if you look at the definitions of both in various ways and interpretation, it will differ. So I think that it is better to look at the various definitions, because I have discussion of the same with Dr. Anil Gupta, you know, that National Innovation Foundation. And while we are sitting at the tea table, every time we are laughing to discuss the defining this innovation and entrepreneur, innovation and invention, right? Anyway, so this is up to all of you, because it is a broader view from invention to innovation.
Thank you, sir, for answering the question. The another question is from our faculty, Dr. Tejas Dhamelia. How you have been designing new scaffold for different activities?
Sorry?
How you have been designing new scaffold for different biological activities?
See, three things. Whatever we are learning from the past, many times what happens that when we are looking in the past and we are trailing behind the other researchers, then sometimes we are becoming me too. And therefore, when we are, when our group and we are thinking, we are always finding for the novel compounds and different scaffolds. There should be small molecules. They should be easy to diversify so that we can have a large range of molecules, and then these molecules randomly tested for the different biological activity or specific activity, and out of that, whatever results come, there will be second generation of molecules. For example, I just tell you that with TAF we have done entire work and 5,000 compound anti-tubercular work. But some of the compounds has shown IC50, wonderful. So we separated this group of molecules, diversified molecules, and we had done bioinformatics on this molecule with Professor Coutinho, and then Professor Coutinho has given us the design molecules. Yes, these are the molecules we can design. Then with the help of National Chemical Laboratory, we designed these molecules, and contrary to our NIH experience, this design were so superb that it was working on the dormant tuberculosis, and we were very much successful, and immediately we have patented these molecules and then published the paper. So this is how things are coming in the different way. When we were looking at the P-glycoprotein inhibitors, we were thinking that there were calcium channel antagonists which are like verapamil to dihydropyridine. They are working on the P-glycoprotein target. But we know that when the efflux pump behavior comes, the anti-cancer drug like vincristine and vinblastine, they were pumped off out of the cell membrane, and the drugs were no more effective. So you require an immunomodulator. So we got a tip that calcium channel antagonist can work, and some paper on dihydropyridine, but people have never worked on the molecules which we synthesize. So we thought out of box thinking that let us design the molecules, but in this molecule we have to see that the cardiovascular activity should be very much reduced, and therefore we tie up with the Zydus Research Center and we requested Zydus to test our compound for the calcium channel antagonism. So when we found that these molecules have no calcium channel activity, we further tested it with our Japanese and Hungarian scientists for as a P-glycoprotein inhibitor. And out of that, one compound has shown wonderful activity. That is, we call it as a DP7. So this is how things that work. Sometimes out of box thinking, sometimes intuition, sometimes the data from available literature. But when you follow the other available literature, then depends many times it happens that you are also going to be me too. And therefore you have to be very cautious while selecting your problems and your design your compound. I hope that I answered.
Yeah, thank you very much, sir. On behalf of Eleventh College of Pharmacy, I would like to thank you for sparing your valuable time for this lecture. As an appreciation and gratitude, I invite Dr. Tejas Damalia to present the e-certificate to Professor Dr. Anamik Shah.
Tejas sir?
Yeah. Am I audible, sir?
Yes, sir.
So, Palak madam, will you continue or shall I?
I'll continue, sir.
Okay.
So, can you please share the certificate
Yes,
with us? So I request Sureja sir to present a certificate to Anamik Shah sir as a token of our appreciation and gratitude for this expert session.
Thank you very much, Anamik sir, for your wonderful lecture.
Thank you,
Please accept.
Sureja, for giving me e-certificate. But I think that for all of us, this is a time for ET or T in real sense. So
Yes,
good luck to all.
thank you, sir.
Thank you very much, sir. Thank you, Anamik Shah.
Majama Sahib?
All well, sir. Thank you very much. તમે સમય કાઢીને આવ્યા.
ના મજા આવી બધાને મળવાની આબાને.
Thank you very much.
Okay, sir.
ખરેખર સરસ મોટિવેશનલ હતું અને it's really good.
Thank you.
Yeah. Okay, sir. Thank you very much.
Okay, sir. Yeah.
Thank you, Anamik sir. Now I would like to invite Dr. Ketan Raj sir, Associate Professor, Department of Pharmaceutics, to deliver a vote of thanks. Over to you, Ketan sir.
Am I audible, Palak madam?
Yes, sir, you are audible.
Honorable Anamik Shah sir, respected principal sir, staff members of Eleventh College of Pharmacy, dear students and participants, let me extend a vote of thanks on behalf of Eleventh College of Pharmacy, Research Committee and IQAC. Now, what we see that we stand on the curve bidding bye-bye to Vikram Savan 2078 and paving the way for upcoming Gujarati New Year of 2079. And we had an absolutely dignified academician, professor and scientist who has summoned a graceful, befitting end note to our webinar series. Anamik sir, we have received an excellent feedback for our webinar series of Science in Spotlight, and where participants have been saying that it is an intellectual feast and whereby they are able to add value to their knowledge. And I feel with this one more webinar of the October 2022, we could take this brilliance even one much higher. So I thank all the stakeholders of Eleventh College of Pharmacy to create this history time and again, as we are also celebrating our 75 years of establishment of the institution. The chemical science has embarked a new elevation since few years because of research and education by Professor Anamik sir. So we are really honored and appreciate, sir, for your inspired presence among us at LMCP platform, and also for delivering a wonderful, thought-provoking, very informative lecture focusing on research and innovations in drug discovery at academic institutes and hybridized collaborative model with pharma industry. So we at LMCP realize that this is something reasonable in current times to be made as spotlight of talking. Collaboration is something the future. And also you have discussed on ethics, what you have inculcated during your education and research career. So thank you so much, Anamik sir, for accepting our invitation to deliver your expertise and experience in this webinar series. So we are truly, we are truly honored for your esteemed presence. I'm also thankful to Chairman Dr. Dipen Bhai for chairing this session in a very effective way, nicely summarizing the lecture and taking up the question and answer session with our speaker. Sir, we are grateful to you as well. So at LM College of Pharmacy, we could do the things in a better way because of our beloved principal, Dr. M.T. Chabaria, sir, a rock solid, strong support. It is his silent keenness and intellect that keeps us on the go. So thank you, Principal, sir, for raising the level of webinar series once again. We are also thankful to all the head of the Department of LM College of Pharmacy for their encouragement, guidance, and support. I am also thankful to all members of Research Committee and IQAC, especially my colleague Dr. Tejas Dhamelia and Dr. Palak Parikh, that we could not have done this without your commitment and untiring efforts beyond the call of duty. We are also thankful to all the staff members of LMCP. This was only possible with your relentless support that we have completed 11 webinars, whereby even one of them was a full one-day workshop. So we are thankful to all the participants for being part of this webinar series. So once again, Anamik sir, we are very much thankful to you for accepting our invitation, and you have joined the legacy of all great speakers whom we have invited since more than a year at LM College of Pharmacy. Thank you very much, sir. Namaskar.
Thank you. Thank you.
Thank you. Thank you, sir. Thank you very much.
Thank you very much, sir, for vote of thanks. With this, we have successfully completed 11th webinar of the webinar series, Science of Science in the Spotlight. Now I request all the participants to switch on their cameras for the group photo. Thank you all. We once again offer our sincere thanks to our honorable speaker of this scientific session and all the participants for joining with us. Thank you very much.
Thank you, Palak Ben, for everything and today's entire thing. Thank you.
Thank you very much,
Thank
sir.
you.
I'm sorry for some technical glitches during the beginning of the webinar from my side.
Give my remembrance to Manta madam.
Sure, sir.
Okay.
Thank you, sir.
Okay. Okay, I'm leaving.
Thank you very much, sir.
Thank you so much, sir.
माझ्या तुमची